send the questions. Okay, there is a reason why it's really good to spend 3 years in junior high AV club. Because later in life, when you have other responsibilities, you actually know how to set up a projector and get your Zoom. You didn't have to do that, but anything. So, hi everybody. I am, do I need to be introduced? I'm just launching right in. Go ahead. Okay, that's fine. I'm Eric Green. I am.
It's like I am gone, James Bond, I'm Green.
That's right. I'm a former director of the National Human Genome Research Institute. There is a story to be told there. I will tell it at the end, and then I'm happy to answer questions. I am late, and it's not my fault. I'm late to the class because for, I think since 1995, if we have it right, Roger? Somewhere around that, I have made it every single year through hell and high water, usually to give one of the very first context-setting lectures in this class, and I've always enjoyed doing it because I've been at NIH at the Human Genome Research Institute since 1994. So I shortly thereafter, Roger recruited me, and I've been giving lectures ever since. And it was all scheduled this year to be an early lecture in the class, and then things happened. But at the time, I was still the director of the National Human Genome Institute. But the Trump administration had us locked down. We weren't allowed to give any talks. And then even when we could give talks, we could only give virtual talks. But at the time I was supposed to give this, I was not allowed to give any talks. Complete ban. And so I was all ready to give a lecture. And then finally, when they lifted all of that, we got this scheduled. But then I became the former director in the interval time, and I'll have some things to say about that. Now, so a few other things. First thing is, because I normally come really early in the class, so I always talk about what I feel I can do to help you sort of get ready for the rest of the classes to set a context, and I'm going to still do that. I probably am adding a little more history than usual because it sort of seems silly for me to go over a lot of things you probably already heard, and I figured, I saw the schedule and a lot of young people giving lots of lectures. They don't give as much history as us old people, so I thought it would be more fun. Plus, I knew that Roger in particular was likely going to show up, and I always like to throw in some new slides. And Dave sometimes comes to these. I like to throw in some new slides to keep them entertained. So I am going to have some new material that I've developed in the last year on the historical front. But we'll talk a little bit of science and certainly a little bit of medicine and even a little bit about societal stuff, because that is the story of human genomics. It has a great history because of great science, increasingly relevance in medicine, with a lot of complicated societal challenges that come up as well. Now, what I gave you was already prepared. It actually, in some ways, is a little odd that I'd be sharing this with you, because I really am not supposed to represent the National Human Genome Research Institute anymore, even though I was there for 30 plus years and I was the director for 15 plus years, but I do not know more. But we just have a really nice brochure. And I guess, actually, I bet you that one pager, actually the one pager I just realized is probably out of date, because, well, it has to be out of date, because it probably says I'm the director, which I'm not. All this was prepared in January, but I didn't think that you guys should be shortchanged just because I lost my job, that you shouldn't get a nice brochure, also a nice pin, which we only made in the last year, which is like way cool. And I'm going to talk about the reprint when we get to it. So anyways, you get all the benefits of having the insurance director here, even though you really don't have the insurance director. So I'm not officially representing the Institute, but the Institute sends its regards. So what I get to do is to tell you about my view and some little bit of history and science and medicine, sort of how we've gotten. I really want to just tell the story of how we've gotten to where we are today in human genomics, which you guys are so fortunate to be training at a time that you are, and especially in a place like this, because it's this incredible history, and I want to tell you the context. I would contend, and I don't think Dave Valley is going to disagree with me, that two scientific fields that were both launched last century really are changing medicine this century, which have drawn me as a physician scientist into this field. For those who don't know the full history, the word genetics didn't exist until 1907. This is the first time it appeared in the scientific literature. That was early on in this century. And then, of course, sort of halftime of last century was this incredible discovery in 1953. And then bookending this remarkable century of naming fields was the naming of the word and the coining of the word and the launching of this field of genomics in 1987. Now, how did we sort of get in this progression, especially to actually creating a new field? That, by the way, the other reason I give more history is every year when I come here, you guys just keep getting younger and younger. I'm staying the same, but the students always look like they drop 3 years every year. And I just know that none of you were around in 1987. So, this makes it easy to want to make sure you know. Now, why was there this remarkable creation of this field of genomics, which you are all now beneficiaries of? There really was this accelerated progress in DNA research that started at halftime of the last century with the discovery of DNA's double helical structure. But then, by the 1960s remember that famous, lookup table that you all memorized in like AP biology class, or at least you know how to use it. This was the genetic code, which came out in the 1960s, and actually most of that work was done on the NIH campus in Bethesda, Maryland. That gave clues as to how it was that DNA encoded the information for making proteins. And then the 1970s, by the time I was in high school-ish, was a time where all of a sudden we were developing the very nascent methods for sequencing DNA. And then the 1980s were really all about DNA manipulation, with the advances in recombinant DNA cloning, which began in the 70s but really blossomed in the 80s. This is when I was a graduate student. When I was at your stage, the rage was a typical PhD thesis, to be honest with you, including some of my MD PhD classmates, was to work hard and isolate a series of overlapping cDNA clones for a gene of interest, sequence that, 1,500 base pairs of sequence, analyze it, and write it up. That's your thesis. Now that's a lookup in like 30 seconds. Any of you could do that. But that was it. I mean, this was the rage of PhDs in the the mid 1980s. But also later came PCR methodologies and so forth. And what that led to was a growing tool belt around geneticists, in particular, and molecular biologists being able to study, analyze, manipulate DNA more and more and more efficiently. And what that then led to was what I refer to as a drumbeat of discussions that had started even before genomics getting coined, where they started to have meetings. This is a very famous meeting that took place in 1984, and then a major editorial that was written in 1986 that really led to this accelerated interest in being comprehensive in the study of the DNA of an organism, which of course is its genome, leading to this coining of the word genomics and the first use of the word genomics in the literature in 1987. Dave would probably want me to point out that this editorial was written by the editors of this brand new journal called Genomics, of which one was Victor McCusick, who's right here, of course, and Frank Ruddle, the two editors, or the three editors?
Tom Roderick. I think Tom Roderick might have been an author on this paper.
Okay.
He came up with the.
He came up with the name over beer in a Bethesda workshop. Yeah, that's the
I think at Bar Harbor.
At Bar Harbor? Oh, maybe. Okay. So, any case, and the reason genomics got named was this idea was starting to coalesce about a big, audacious project that maybe could tackle the mapping and the sequencing of the human genome. So they started to get much more serious around the time that the genomics word got coined, with major groups like the National Research Council and other groups getting together and publishing a blueprint for what this might look like. That then led to major congressional hearings in 1989, which is what then resulted in Congress allocating the money and creating an entity at NIH, as well as giving some money to the Department of Energy to launch the Human Genome Project. So, 1989 was an incredible year. Now, let me point out, I graduated with my MD PhD here, and I was just, sort of extremely fortunate to make decisions about where to do my postdoctoral work, and landed in a pioneer's lab in genomics, a guy named Maynard Olson, that allowed me to, starting in 1988 as a postdoc, to really start using the tools that would eventually be applied to the Human Genome Project. But 89 was an incredible year. And I can remember it vividly. First of all, just everything changed in 89. Let's just get started with really the most important, Taylor Swift. We all remember that. Okay. But, it was also the countdown year for launching the Human Genome Project. So again, you have to throw some nuggets in for Roger, or he's off. So you have to throw things at him that he's never seen like this. So what this led to, of course, was this launching of this project called the Human Genome Project. I was fortunate; I was in the right place at the right time. I was at Washington University. They got one of the first major genome centers, along with about 5 or 6 other places. And literally, as a postdoc, I was given the ability to work on the front line, mapping and sequencing, well, particularly mapping initially human chromosomes. I think all of you know bits and pieces about the Human Genome Project. I do find as I lecture more and more to audiences that look younger and younger, it gets less detailed knowledge about the Human Genome Project. And I know I'm sounding old, but this was really historic. And I think it's important to, you have to respect the history, you have to respect the shoulders you're standing on. And you are really standing on the shoulders of a remarkable, audacious project. It was so unusual. You were all students when consortium-based science, big data, that's all the norm. This was the first foray into that biologist. It was, not an idea that everybody was thrilled with. It was all about international, but especially today. I mean, holy cow, I don't know how we would do the Human Genome Project today because there's such an anti-international, wouldn't get funded going on. So, this was, and it was big science, it was managed science, and I would also really point out, it was, not a sure thing that it was going to succeed. Now, why was it not a sure thing that it was going to succeed? So here's a new slide. I love this slide, Roger, so I want you to really enjoy it. I went through.
My seat here.
What?
This is just fabulous.
Okay. I want to remind young people, as much as I can, about what did not exist at the launch of the Human Genome Project. So just think, we are doing one of the most audacious scientific endeavors in the history of humankind, and yet this was what cards we were dealt when the Genome Project launched. We did not have web pages. They came in 91. We did not have texting. That came in 92. We did not have live streaming in 93, nor any browsers. They didn't exist until 3 years into the Genome Project. PlayStations, no GPS until 95. No Amazon until 95. Windows didn't even exist until 95. USB cables, DVDs. No Wi-Fi. Believe me, no Wi-Fi. And Google wasn't even available. No Google searches. Social media didn't come until 2000. So we said, we're going to map and sequence the human genome. We have none of these things that we all take for granted today were available to us. By the way, things that became available after the human genome, so it's now 2003, all of these things, which we rely on every day. And yet, none of this existed throughout the Genome Project. Okay, now you may say, what kind of society existed without these things? This must be incredibly prehistoric. Yeah, we did have film cameras, and film cameras were very good at capturing us, either working on the Genome Project or, in my case, arriving to work on the Human Genome Project. There you could see. Now, let me tell you some other realities. Oh, by the way, here's a picture of me. So here, this is, so first of all, you can see I was once upon a time roughly your age. That's the first thing. And this is literally a piece of the human genome. These are all, the horizontal lines are yeast artificial chromosomes, each about 200 to 800 kilobases in size. Each of these little markers along the way are landmarks along human chromosome 7, which I was responsible for mapping in the Human Genome Project. And this was the first generation physical map of the human genome that was done for, in this case, chromosome 7 by the Human Genome Project. Let me point out some additional key realities besides the fact that we lived in prehistoric times without iPhones and web pages and browsers. By the way, there were only, I think, it was one other thing, I didn't make a slide of it. There were only, at the time the Genome Project launched in 1990, there were only 3 million people in the world who had access to the internet. Just imagine, only 3 million people in the whole world had access to the internet when the Genome Project began. There were some other key realities. The field didn't totally exist. I mean, the field was named in 87. It was a toddler field. Nobody in 1990, when the Genome Project launched, nobody raised their hand and said, I'm a genomicist, because nobody really trained as a genomicist. I mean, I raised my hand, I said, well, I'm a cell biologist and I'm a pathologist. That's the best I could do. Dave raised his hand, he said, I'm a geneticist. But it was a melting pot of people coming together from physics and from chemistry and from biochemistry and et cetera, engineering and so forth. The second thing is, I hope, even though they had that drumbeat of discussions with lots of documents, there was no detailed start to finish plan for the Genome Project. There was the furthest thing from the truth. I mean, it was all about just figure out what you're going to do next and get it done. It was absolutely like building an airplane while you're flying it. This was absolutely what we felt like most of the time throughout all 13 years of the Genome Project, and just keep going, just keep going. You know, we pass on vomit bags because it got turbulent. And there's lots of anecdotal turbulence issues along the way that I'm not going to tell you. But the other thing, and here I'm going to insert something new that we just did in the last year that you might be interested. Sometimes I think people don't realize, I mean, the Genome Project, look at it, I'm going, duh, of course. I mean, how could, of course, this was like, what a great thing. It's created this whole field. It was not so popular. There were mixed feelings. I mean, there was some supporters, but there were mixed opinions. And so when I was the NHGRI director, I created a history of genomics program early on, and we had a staff historian, and et cetera, et cetera. And we've been creating all sorts of digital assets for educational purposes. And so in the past year, part of the reason we created a history program is we had just reams and reams of boxes of documents, all wonderfully historic and insightful about the Genome Project and the time since. But we created this one digital exhibit we talked about by one of our archivists who basically cataloged a writing campaign that happened that you'll think is really interesting. So I'm going to share this with you. So I'm just going to, this is if you want the digital exhibit, which you find by Google, has a lot more letters and a lot more material, but I'm just going to give you a sample, just to give you a sense of what life was like when the Genome Project launched. So in June of 1990, the Genome Project launched in October 1 of 1990. New York Times ran an article talking about the Human Genome Project and mentioned how the Human Genome Project is bad, quote, the Human Genome Project is bad science. It's unthought out science. It's hype science, says Martin Rechsteiner, who was a biochemist at the University of Utah. Some critics have begun aggressive letter writing campaigns urging colleagues to harbor similar sentiments to write counters. And so Martin actually...
Just interject here. Martin Rechsteiner is a graduate of the biology department, PhD from Johns Hopkins.
Okay. And you can all decide when you're done with this whether you think that fact should be publicized. Because what Martin did, actually I know people know him, they said he's a nice guy. What Martin did is he did indeed write this very lengthy letter, a very long letter to lots and lots of people in February, earlier in the year, where he said, if you agree with the arguments presented by Martin about how bad it's an idea, you should write science advisor Alan Bromley, NIH acting director William Robb, then Senator Al Gore, or Senator then, who's no longer with us, Ted Kennedy, and express your reservations, concerns, or opposition to the Human Genome Project. Kennedy and Gore were big fans. They were also on the Senate committee that gave money for the Human Genome Project. And he then went on to say, in short, the Human Genome Project is a waste of national resources and is detrimental to the training of young scientists. I urge you to take the time to voice your opposition. 5,000 letters would have a significant impact. He got about 45. But we have like all 45 of these letters. So let me show you what these letters are. And by the way, I want you to realize this. Just imagine if you were a trainee working on the Human Genome Project and some people are dedicating your career, as I was as a postdoc, and people are writing letters like the following about this project. So this is somebody from the University of Minnesota who writes, the Human Genome Project is a costly, wasteful, and inappropriate allocation of research funds. The Human Genome Project will provide little useful training and no intellectual stimulation to young scientists. Finally, here we go. 95% of DNA does not code for proteins and is thought by many, including advocates of the Human Genome Project, to be junk. A famous four-letter word. They always talk about junk in the non-coding parts of the genome. Here's somebody who breaks my heart because he's from University of Wisconsin. Where is this right? Department? I think this is right. I was an undergraduate. I am certain that most of the sequence information that will be generated will never prove to be of any scientific value, especially since probably greater than 90% of the sequences are non-coding or junk DNA sequences. To which I say you can have a junkyard, but even your naive view of what might be junk might someday actually have some value, so I actually think that's completely not true, and he proved it. The Human Genome Project is a brute force and indeed stupid way to determine those sequences that will be of biological interest. They didn't like this project, did they? And here is, that was two of the letters. There were several, there were a bunch of others. But then there was also this review article, not part of the letter writing campaign, which you can't believe that that these people said these things during the genome. Michael Waterman, who's a good guy, wrote this feature where he talked about, while drinking brandy after dinner, Wally Gilbert, who was the inventor of one of the two methods for sequencing DNA in 1977, wondered how to find the labor for the boring, repetitive sequencing. Gilbert proposed that we use prisoners to do the work. This is a true story. This was the room that they thought it shouldn't be trainees like me, it should be prisoners. Give one group the Crick strand and the other group the Watson strand, how to create competition and quality control. I knew that Gilbert had no idea what he was talking about. So I could just tell you that this was the attitude back then. I was just trying to map and sequence the human genome, but I was being told this was more like what I was. And I remember, in the face of this, one of our key leaders was Francis Cowell, so there you see Francis standing next to me. And I remember going to Francis once and saying, Francis, they're all being so mean. They're saying such horrible things. They don't like this project. They hate us. How do you get through this and stay so optimistic? I need some cheering up here. And Francis looked at me and without missing a beat, he said, the hater's gonna hate. I shake it off. It was the first time I ever heard that. I think she stole the lyrics in the Genome Project, but I can't prove it. But I'll leave it at that. Okay, but those letters are real, and that review article is real. The Taylor Swift thing is a joke. Last thing I should point out about key realities of 1990. I can't stress enough how crazy it was that we were going to try to map and sequence the human genome without a functional internet. It was just assuming there'd be way to distribute the data. Why do I say that? When the Genome Project began, this is how you used to distribute sequence, in books. Oh, it did get upgraded a few years in. It went to CD-ROMs. GenBank was not distributed on the internet. GenBank was distributed on a series of CD-ROMs. First one CD, and then two. It became a box set. And when it got to be a big box set, they said, all right, we got to put this up on the internet. Literally, you'd get in the mail. What was it, Dave? Like every 3 months, every 6 months, you'd get updated. They would mail. I mean, it's just, you can't even imagine that's how you do it now. Last thing I would point out is the earliest, when I was developing the map of chromosome 7, the earliest sequence data that was being generated on my behalf from some flow-sorted chromosome 7 samples over in New Mexico, so that I could make PCR assays to them to use these as tools for identifying landmarks in the DNA. At the time, there was no internet. This literally is 1990. And so my collaborator would get a little bit of sequence data. He would handwrite it, read it off the sequencing gel, and he would fax it to the Department of Genetics fax machine. And it wasn't even a plain paper fax machine. It was one of those thermal things where it was a roll, and then it would roll in. And I would manually look at it and design primers by eye. This is how we started the Genome Project. True story. Truly did use fax machines for distributing some of it. All right, so now I've said all these things. I hope I have at least elevated your respect for the Genome Project, recognizing the headwinds that we faced when it first began. But we did get it done, and we successfully completed the project in 2003, even ahead of when we originally thought it was going to be. And there we have it in front of us, and I then started immediately reading the human genome sequence, which was located in a book that looks something like that. I do like to point out that I talked about how incredibly historic the Genome Project was. I think the only thing that's analogous, and this is, I know long before you were born, because the moonshot took place when I was only like, a 6 or 7 year old or something like that, or I guess, 8 or 9 year old. And, putting a person on the moon for the very first time. I mean, it was a big project. It was this big, expensive, large managed project. And its signature goal, that is putting a person on the moon, has been repeated 11 times. I mean, I know I'm biased, but the Genome Project, it was a heck of a lot. But biology's equivalent of a moonshot. It was a heck of a lot cheaper. And you know what? We've now repeated it over 1 million times. So just saying, I have great respect for this. but we've reduced this to practice, as we're going to talk more about. And it's been done many times. But I would certainly contend, I think these are the two most incredible scientific endeavors of my lifetime. But what I want to move on now and talk about, that was a little bit of the history lesson up to the Genome Project, but we have to appreciate the fact that the Genome Project's completion, now 22 years ago, it was a finish line, but it was just as much a starting line. I mean, it really was just a start. We fully appreciated that the Genome Project was just going to be foundational, and all the excitement would come from what you were going to do with this new information. What happened 22 years ago was sort of a scientific diaspora, if you will. I mean, genomics went all over the place in the life sciences, as it should. Whether it was agriculture, ancestry, livestock, infectious disease, forensics, I mean, all of these areas are, you can almost have entire courses around them now, just covering material of how genomics accelerated advances in these in the last 22 years. What we did at the National Human Genome Research Institute, it became an identity issue, right? Because we were created by the US Congress as one of the NIH institutes to lead the Human Genome Project. And of course, with the Genome Project done, some people say, well, do we still need this institute? And of course, we said we did. And there was good reason for it, because we recognized that a huge growth area needed to be the application of genomics to health and to disease and to medicine. And we were poised to do that at the NIH and really leading on it with a heavy focus on the genomic aspects of it, ready, and also critically important, making sure that we then expanded the use of genomics across all the other areas that the NIH was focused on. So our focus was going to be pivoting to the applications of genomics to medicine. Now that wasn't that radical of an idea. I will tell you that when Congress was convinced in 1989 to fund the Genome Project, among the arguments that were made by physicians. I mean, there were scientific arguments, there were a lot of basic science, but some of the arguments that I believe got some traction was this idea that we knew that each of us was a little bit different, and yet the differences that are encoded in our DNA, clinicians were blind to. And we needed to open that window someday so that physicians and other healthcare professionals could take advantage on those aspects of our DNA differences that influence our health, but they are blind. And so, but just imagine a day that if we first understand the human genome, could we eventually apply it to individual patients? And that was a dream. So that was always the bubbling of the background, the health applications of genomics. So none of us were terribly surprised that the juxtaposition of the two words genomic and medicine even came before we had time to even make it. I mean, New York Times picked this up right away around the time the Genome Project ended, as did a magazine or a journal like Science Magazine. And so there was already this great interest of thinking about how genomics can be applied to medicine. There was a slight problem with this 22 years ago, is that we could spell genomic medicine, but it was a very blurry concept. I don't think any of us really had an idea what it was going to look like, and we knew that one of the most important things was to sort of step back and think about how we were going to eventually bring clarity to this, recognizing this was never going to be simple, but that this was something that clearly had to be a high priority. Now, we can start to define it. I think we started to appreciate that genomic information, variants in our genome, et cetera, about individual patients probably could be very helpful for at least understanding the components of human disease and human health that are scripted in our DNA. There's obviously lots of other influences on our health, including physical and social environment, but let's at least understand the genomic side of it. And so, we could imagine defining it, and then we just have to take a step back and appreciate that this pivot was going to be another one of these long journeys, right? Again, the starting line was going to be the end of the Human Genome Project, and just like we had a previous finish line of a sequence of the human genome, now we were going to have to realize genomic medicine. Now, we also, I think, were very realistic, going back to the strategic planning that went on in the end of the Human Genome Project, of recognizing that this was going to be probably we knew it was going to be much longer than the Human Genome Project. We knew it was going to involve a lot of steps. And just like the Genome Project, we knew we wouldn't know all the steps when we started. But what you just got to do, just like mapping and sequencing the human genome, you say, what do I got to get done first? And then if I get that done, what's next? And then you have to be gratified when you accomplish that and you check it off your list. You say, all right, got it done, now I can go on to the next thing and the next thing. And I would really categorize our last 22 years as being this exact high-level graphic of what we needed to accomplish. But the only other thing I'd say about it is we also appreciated that just like the Genome Project, it's not going to be one scientist, one country, one funder, one discipline, and it was not going to be a sprint, it was going to be a marathon. It was going to be lots of people, lots of disciplines, lots of countries running shoulder to shoulder and figuring this out, and we knew it was going to take probably multiple generations. So how do you go from having this idea of what this is going to look like to actually operationalizing it, especially if you're a funder? So again, in my previous life when I was the director of a funding agency, I can tell you that what the Genome Project did was rely on regularly developing strategic visions and publishing them, being very transparent about it, bringing the community together, strategizing, arguing, even fighting, and coming up with documents that would be helpful. And I will tell you that has been embedded deep in the culture of the human genomics community, I think led by the Genome Institute, where we have taken that same approach, starting the day the Genome Project ended, to put out a strategic vision, and then when that vision became a little bit in need of updating, putting out a new one, which this one was published just as I became, when the baton got passed from Francis Collins to me as the director of the Human Genome Research Institute, and then our most recent one was completed, actually in the middle of the pandemic. Each of these represents about 3 years of strategic planning with the community, and then you get it all, try to get it down to about 10 pages that fits in a Nature article. In the back pocket of the brochure I gave you is the strategic vision. I'll have a few more things to say about it. But that's the latest strategic vision, which is now 5 years old, which I can hardly believe, but time flies. So I think what you've heard bits and pieces of in this class have been many of the things that were strategically being planned across this interval of time. So I'm going to do a flyover and just briefly tell you what I think, and many people might have a different way of organizing this, but what I think have been the major steps that have gotten us closer and closer and actually starting to realize genomics and why. And just, again, it's all about context setting. So I'm not going to tell you anything you don't know, but I just want to sort of have you see the vision of what this journey has been like. The first thing that we absolutely needed to do when the Genome Project ended, and we called out for it in our 2003 strategic vision, and said we need to just get the cost of sequencing much lower. And we've done it. In fact, we've done it by reducing the cost by more than 1 million-fold. It has been truly historic. I want to remind you that when we sequenced that first human genome by the Human Genome Project, it was done in factories that look like this. That is why the first human genome sequence cost about $1 billion, roughly. About $1 billion. And we knew that was too expensive for a clinical test of an individual patient. And we knew it wasn't just about knocking a couple of orders of magnitude off. We needed to knock about 1 million-fold off, another 6 orders of magnitude, and deliver all new technology. And that's been game-changing. In fact, I think it's one of the most remarkable technology development efforts co-contributions by the federal government, but also the private sector. And lots of going back and forth, and whether it's Illumina, PacBio, Oxford Nanopore, blah, 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 Ultima, there's just more and more. It's just wonderful to see. And all this is, it's just truly something that we should be remarkably proud of, that we've accomplished. And you are all beneficiaries of it, because if you can sequence a human genome for less than $1,000, you can do a lot of sequencing, including if you're going to look at smaller model organism genomes, you can really go crazy. So that's been game-changing. What that means is that with cheap sequencing, we have now allowed ourselves to really go at this and sequence a lot of genomes. Nobody even knows anymore how many human genomes have been sequenced. Sometimes we say, do you have a number? It's impossible. It's impossible to know that number. You could look at any given database and say how many are in here, but there's a lot of private data, a lot of companies, a lot of countries that aren't sharing data. There's also a lot of differences in the quality of the genomes, whether they're exomes or whole genomes or light whole genomes or quality whole genomes or telomere, et cetera, et cetera. It's impossible. It's in the millions. But what's most important is that it wasn't so much just to get all those genomes. The idea was to get lots of genomes from lots of different populations to increasingly get as much ancestral diversity captured as possible. I guess I can make one political comment. we're working really, the people at NIH now are working really hard trying to preserve the notion of the importance of ancestral diversity in genomics, trying to explain that to the current administration. They see diversity and they get very concerned about it and cancel grants and all that. We must preserve the importance of getting ancestral diversity in our genomic data sets for reasons that all of you are very familiar with. And that has been really important as we've sequenced more and more genomes, so we can check that off our list. Because the other key thing, and again, this is I'm sure covered in this class, I just can't stress enough that as we've gotten incredibly rich catalogs of genomic variants, of which now we know about literally hundreds of millions of places in the human genome where there are differences that exist in the human population, that when we now project to any given individual, where any given individual has something like 3 to 5 million variants in their complete genome compared to any other reference or any other person. But the problem is we want to focus this down to the small subset that are medically relevant. Of course, we're interested in the biological relevance. We know the vast majority are not at all relevant. But in order to do this, we need to have very facile ways of knowing variant by variant which ones are biologically consequential and which ones are not. This is certainly a very important cutting edge aspect of what's happening in human genomics today. Required to be able to do that, of course, is before you could even know whether a variant influences a genomic function, you must know how the genome actually functions. And I would contend that we've made profound advances in the first 22 years of understanding how the human genome functions. But we need you helping us, and then we need your graduate students and your children, and then probably their graduate students. This is going to be multi-generational. Those 3 billion letters are really complicated. And all that junk that they kept calling it, some of the most interesting and rich biological information living out in non-coding DNA, and they called it junk because they didn't understand it, and we still don't totally understand it, but we know it ain't junk. We know it's incredibly important. And so I would contend this gets a medium check mark because I think we have a long way to go. But I think it's you live in a world now where you can view the genome in browsers like this. I always make the analogy that it's basically a Google map. And instead of looking for hotels and restaurants and bars, you're looking for expression of RNA, conserved sequences, non-coding RNAs, genes, and so forth. And it's getting better and better all the time. But we got a long way to go. And then, of course, once we have anything approaching knowledge of it, we have to appreciate that there's far more that we don't know than we do know. And the goal here is having talented young people like you working with great people like at Hopkins, just keep pushing on this. Just get more and more of it above the surface of the water, making more and more of it known. And that's so critically important because when you want to juxtapose all this variation data on all this functional data, you just have to have really strong functional data to have any hope, especially on non-coding DNA, but even on the coding DNA, have any hope of being comprehensive in understanding which variants are biologically and medically important and which ones are not. And I'm sure many of you are working on trying to push this iceberg higher and higher, as maybe you're working on gene function, just trying to get a better understanding of the function of all 20,000 genes. A lot going on in this campus, I know, in the epigenomic space. RNA structure and function, obviously transcriptional regulation, proteomics, and there's other areas as well that we need to be pushing up on. So, of course, each of these things that we've named so far are really on the very basic science side of this journey. Still working on genome sequencing, variation, genome function. If we're going to really start thinking about how this is going to be applied to medicine, we need to start thinking about human disease. And of course, poised to take advantage of all this was this incredible momentum of the entire human genomics community. But remember, they had about an 80-year head start. They were ready to go to try to help use these new tools of genomics to better understand the genomic basis of disease. And I think there's, in 22 years, been significant advances on unraveling. We aren't totally there. That's still a medium check mark. But I know during this class, because I looked at your syllabus, I know that you had exposure to experts thinking about both rare genetic diseases and common genetic diseases. And I realize there's an imperfection. These are not dichotomy. There are things in between. I'm not going to go through this. I know you guys are experts in this. Of course, the real successes so far have been in the arena of rare diseases because they're more simple, because you're looking for one gene. If you didn't know this number, and I'd like to give numbers to highlight the Genome Project contribution. When the Genome Project began on October 1 in 1990, there were 61 rare diseases we knew what the gene was. 61. Two of them were sickle cell and cystic fibrosis had just been discovered, so those are two of them, sickle cell and cystic fibrosis. But now it's either at or nearly it's either nearly 6,000 or maybe we went north of 6,000. That's pretty incredible. Now, there's probably about 8 to 10,000 genes of Mendelian disorders, so we still have a ways to go. We have efforts going on to conquer those, but that's pretty impressive, going from 61 to about 6,000. Now, you all can appreciate that common diseases are more complicated, much more social, physical, environment influences, many more genomic variants, not a single gene, unlike rare genetic diseases. And of course, the entire era of genome-wide association study, GWAS, giving us literally hundreds of thousands of associations between genomic variants and health-related traits in not all cases. In fact, in only a few cases have we actually gotten at knowing what the gene is that's responsible, what the influence is. It's given some valuable insights, but this is a hard problem. So I would contend this is looking really good, although the remaining unsolved Mendelian cases are intriguing, and I'm sure there's something new we're going to learn as we push harder, and this has to be a big priority, especially when you have administrations that really want to emphasize chronic diseases, which actually is a perfectly reasonable idea. We are trying to communicate, or my ex-institute is trying to communicate with the new secretary to point out that genomics has a lot to contribute if he really wants to understand the chronic disease epidemic, as the Secretary of Health and Human Services, RFK, talks about. And wants to make this a priority area. I think genomics has a lot to contribute.
I like to mention that genetic disease is the most chronic disease.
No, that's a good point.
Begins at fertilization.
Yep. And I'm not convinced Robert Kennedy totally gets that.
I haven't had a chance to speak to him personally yet.
So, but what I will tell you, when I first became the director of NHGRI, and I really pushed hard that this was the journey the Institute needed to do, there was a little bit of pushback that some people said it was, you're really not going to see genomic medicine anytime soon. You should maybe just emphasize these first couple of steps, not try to get all the way to the other side. And I'm glad that the majority of people, among them I will point out, because he was on my advisory council, was Hal Dietz at the time, really was very encouraging to push hard and stick by my guns, which I did. And I think what that's led to are some vivid examples of genomic medicine now emerging, which is really incredibly gratifying for me because I wasn't sure how soon I would see that. I'm going to have to give this a small check mark because I think it's just the earliest days. And you've heard about some of these things, and either in this class or elsewhere, I'll just list them. But what finally came together was this idea that you could take a reference sequence and then you could sequence a patient's genome now for less than $1,000, and you could compare them and come up with your list of genomic variants and put out a report and then implement genomic medicine. Now, I'll come back to this slide because I admit it's an oversimplification, but just go with me for now, and I promise we'll come back to this slide. But we were starting to see this in certain areas in medicine. I will tell you that what I have found to be interesting in watching genomic medicine sprout up in different parts of the medical ecosystem is that it was interesting. We didn't have a grand plan of where it was going to be when. It really was, in each case, a little bit opportunistic, that just was the right thing at the right time in this little medical area. Some of it was predictable, but other areas weren't. And so let me show you what I think is the current highlight list for genomic medicine. The first, I wouldn't even want to say more than one sentence about while standing on this campus. Cancer genomics is incredible. The Hopkins is an epicenter for excellence in this arena. I contend that it's clearly here and now for some cancers, that list will grow. I actually think in the long run, the greatest impact of genomics in medicine will be in the arena of cancer. And you have many experts here to talk to, and it's just remarkable, and just watch it grow. Second area, also prominently featured on this campus, rare genetic disease diagnostics. I have to tell you, when I trained in pathology and diagnostic medicine, I never thought in my lifetime you could actually, especially before the Genome Project began, that diagnose a patient's condition by sequencing their genome. I mean, this is crazy. Imagine that that's happened in my lifetime. I don't know if, David, you ever thought you'd see that in your lifetime, but we're seeing it every month, dozens and dozens and dozens of times, right, hundreds of times around the world. It's happening in routine cases of just the traditional workup of a medical geneticist. It's happening in niche cases, like rapid sequencing of acutely ill newborns in neonatal intensive care units. This is catching on, in many, many countries already, where they are now getting rapid sequencing of, in some cases, every newborn that gets put in an ICU. In some countries, they're just sequencing every genome. That's just incredible. The fact that you can do it in 24 hours is remarkable. The other place this has been seen and widely applied has been in the arena of undiagnosed conditions. People have been on diagnostic odysseys, in many cases adults, for decades, no diagnosis. Bring them in, find a big workup, but also including a genome sequence. You actually get answers. Sometimes you get treatments, but at least you get answers. So this is absolutely mainstream and is just a complete success story in genomic medicine. Non-invasive prenatal genetic testing, this small amounts of fetal DNA that float around in maternal bloodstream. It's the #1 genetic test by sheer number. It's estimated that 8 million pregnant individuals will get this screening test done. I just talk to people all the time, left and right, relatives, friends, kids of friends. Then this is just offered. And long gone are the days of routine amniocentesis, chorionic villus sampling. This is just now. And a part of the reason why the uptake has been so spectacular as the #1 genomic medicine test is because insurance companies leaned in and started paying for it. The second you get things paid for, all of a sudden it's big. That's a big battle that we constantly face. So that's another real success story worldwide. Pharmacogenomics gets talked about a lot, and there are success stories. There are clearly need for more consideration of DNA variants in individuals to select the best medication, so we could be a little more precise in how we are prescribing medication, both the selection of which med and also the dosing. A lot of great research has been done here and a lot of good evidence of it being effective. It's been slower than we anticipated, in part for a couple of reasons, from what I understand. In part, slowness of insurance companies to pay, and also making it more push button and to get it into the clinical workflow of very busy healthcare professionals. There's a lot of efforts going in to use electronic medical records and clinical decision support tools with pop-ups and things to make it sort of a more convenient thing. And I think we'll see over time. I'm hoping that there'll be more evidence for more drugs and make it a little more widespread and get a little bit more momentum. But this has been an area where there's great promise and uptake has been slower than we anticipated. Last area, sometimes people say, oh, I talk too much about medicine, what about prevention? And I sort of blur those lines and maybe I'm imprecise in my wording. I mean, I think there's lots of very good examples of genomics-based prevention, starting with conditions. Now we know that sometimes we pick up by newborn screening of dietary needs or dietary changes that are needed based on genetic lesions of genes that are relevant for certain metabolic aspects of individuals' diets. Simple diet change can make a big difference. Or the kind of screening that goes on in families where you see a lot of colon cancer, then when you look for things like Lynch syndrome, you do really a lot of advanced surveillance of these individuals genetically at first. You find people carrying any of these mutations that make them a higher risk. You could intervene with more aggressive colonoscopies, same with breast cancer, for mammography, and so forth. It's real. I mean, the list is just growing of routines that are now used for genomics-based prevention. So what I would say in this flyover of 22 years of progress is that just imagine where we have come from the earliest recognition that the two words genomics and medicine belong together, and now it's much clearer. I don't make any promise. I don't make any claim that the examples I gave you are even the examples that will be the same even a year or 2 or 10 from now. They're just the tip of the iceberg, and we could all talk about and maybe some of you are working on things that might be the next big advance in genomic medicine. It's a small check mark, but boy, the momentum is there, and I think you're going to see that list grow over time. So let me just now sort of make you, remind you that everything I've discussed, starting with the Genome Project and then this pivot to genomic medicine, made these linear lines, like this is a journey that's all straightforward. You just keep walking down the path and you get to the destination. And that is misleading. Nothing about the Genome Project, nothing about movement towards genomic medicine is linear or without its twists and turns. This is, as you know, this is what research is all about. You're constantly getting thrown in a different direction, and you just have to keep going. And so I want to also give you a couple examples of it's not all simple, and there are big challenges that we constantly deal with. We dealt with them in the Genome Project, starting with the critics of it, but even in the middle, a lot of turbulence. And now I would certainly say there's a lot of challenges we constantly have, although the types of challenges have changed a little, and as I'll get to in a second. But I could give a whole talk on, and it's really what the community thinks a lot about. It's also what the funders and National Human Genome Research Institute and others at NIH think about. Because what our real role is to think about what are the barriers to getting to progress, and then what could we do to get these barriers knocked down. You know, if you ask me 2003, what was the biggest challenge? Well, the biggest challenge was the cost of sequencing a genome. If you ask me today, what are sort of the big challenges, especially in genomic medicine implementation, I'll give you a couple examples. Only for time reasons, I can give you more. But first is, I oversimplified how it is to take a genome sequence and make it clinically useful. I warned you that this was an intentional oversimplification. I reduced into one slide what is an entire field of study that some really brilliant people are working on, but it is really hard for a variety of reasons. And the truth of the matter is, yes, you know, we could go into the hospital here and yes, we could sequence a genome if we wanted to rush it, we get it done in a day or two, and we can get that list of 3 to 5 billion variants. But you know, when Dave goes and rounds on that person in the morning, that's what Dave feels like. You know, most of the variants, we don't know if they're relevant or not. Yes, we cream skim and get answers in a lot of the cases. But the vast majority of variants, we have no idea what their clinical relevance is. And it's a combination of complexities, but it really is heavily tied to understanding the aspects of ancestral diversity and aspects of then the interface between the DNA variation and genome function, and then of course understanding the underlying pathophysiology that might result from it. So when you go to analyze a patient's genome, first thing you need to do is to accurately get the list of 3 to 5 million variants. And keep in mind, this is why we need the pangenome. This is why we need lots of reference genomes. Because if you're going to look at my genome sequence, you need to have an appropriately ancestrally matched reference genome to accurately find my variants. And if you take somebody else, a very different ancestral background, and you use that same reference that you use for me, you might not even get an accurate list of the variants. So the first thing is that's why we're creating hundreds of reference genomes from people from all over the world, different ancestral diversity, just to get that number. But that number is accurate. When it's accurate, it still was overwhelming because, of course, nobody can analyze 3 to 5 million variants in a patient. So what you have to do is to go through and filter and prioritize and just successfully knock these numbers down. We do this by looking at databases where we've aggregated variants from different ancestral groups. And if you're looking for a rare genetic disease and you see a variant, and that variant is a high percentage in the population, well, if it's a high percentage, it can't be associated with a rare disease, you knock it out. So it's this whole filtering and prioritizing process that becomes critically important. And this requires the development of all sorts of resources, some of which you are likely using, but if you're going to get it accurate and you're going to end up being able to sift and prioritize and filter, you need to have these tools, all of which are being worked on. Just to review, you need an appropriately matched human reference genome sequence or a pangenome, a major program that NHGRI continues to push on with international partners. You need reference population databases, the most famous of which is NOMAD, where you could look and see, all right, I have a variant, is this rare or not? Could this be related to human disease? If it's very common in population, probably not likely to be cause of a rare genetic disease and so forth. And something I'm particularly proud of that we launched shortly after I became director, and maybe some of you are using, you have to have a really good knowledge base of curated information about the likely pathogenicity of genomic variants. And ClinGen is a program that NHGRI and multiple institutes at NIH now support and really has been a major, well-recognized, well-respected group and consortium, a resource actually, for cataloging as much as we could possibly get from the literature and from experts about which variants are relevant in which given diseases. So this is why all of this is active research that is critically important to help us better understand the clinical relevance of variants in managing patients. The other big challenge I'll tell you about is a societal one. I could vividly remember when the Genome Project started, and it was so much fun because we were just a bunch of nerds mapping and sequencing the human genome. And a lot of people didn't really pay that much attention to us at the time, and they just let us alone. And because what we were doing was so circumscribed, there were some things we had to be a little bit aware of, but it was a lot like being at the kids' table at a Thanksgiving or a holiday. I mean, it was just, we were just a small community, just doing our own thing. We didn't have any real big responsibilities. That was back then. What has happened over the last 22 years in particular is as genomics has found its way into medicine, there is simply nothing more complicated in society than medicine. And you see it every single country struggling with how to get good medical care to the individuals. And so what it means is that nowadays, I'm here and I'm being told, no, Eric, you can't sit at the kids' table. I don't care that they're talking about iPhones, Instagram, and Taylor Swift. You have to stay here and talk about mortgages, life insurance, taxes, politics, and genomic medicine and healthcare. This is absolutely what the field is dealing with now, is that we want to be talking fun stuff, but, we got us to this genomics and medicine, and that is just like mortgages and life insurance, tax, and you just got to be at the adult table. And it's really, it's been an amazing recognition that we have to tackle societal challenges associated with genomics just as much as we have to tackle the problem of understanding genomic variants, is that you can't ignore it. It's a key thing of what you do. It's part of the reason why we have an ethical, legal, and social implications research program for many years called the ELSI Research Program, started when the Genome Project began. Congress requires that we spend part of the money at NHGRI to actually study the ethical, legal, and social implications, and boy, it's more relevant than ever. Because when you're at the adult table and you're talking about genomic medicine, all of a sudden, you're talking about how genomic medicine gets delivered. You're talking about how genomics gets paid for in medicine. We didn't have to worry about that stuff before, and now it's complicated. And every country does it different, every health system does it different, and so forth. You have to deal with the FDA. You got to deal with how things are being regulated. You have to think about equity. You don't want to exacerbate health disparities through genomics. All this is an adult problem. And it's the problem for everything that's ever touched medicine. It's not surprising it's going to be a problem for genomics as well. Lots of concern about genomic privacy, just like there's concern about the privacy of genomic information. Oh, and of course, we need to think about literacy. Literacy for patients, literacy for healthcare professionals, literacy for all the people like my medical school classmates who never heard the word genomics once in medicine, in medical school, because it didn't exist as a word. They're out there practicing still. And people are coming to them with genomic information. They're not literate about this. They haven't had time to be. So it's really a huge set of responsibilities that we tackle. And then, of course, there's many others. Many others. We can talk about it afterwards, but we are, needless to say, lots of things I could cover with this. So just in the last few minutes, I just would point out, one thing is the other seismic change I've seen in genomics of late is that I have come to appreciate, especially when we went to put together the latest strategic vision that I shared with you, that's now the one that we're operating off of, is that the big transition in genomics over the last 10 years in particular is that genomics is everywhere. And I just thought it was interesting, so I sometimes show the slide, that The Economist, when they were celebrating the 20th anniversary of the completion of the Human Genome Project, The Economist, not a scientific piece of literature or magazine, The Economist talked about how genomics has changed everything. I just like this quote because it'll make a point. No part of biology is untouched by the consequences of the Genome Project. Genomics has therefore come to form a framework for biology in the way that the periodic table forms one for chemistry. It touches everything. And I say that because when we went to do, and it's even now an issue, and I think it'll always be an issue for NHGRI, they go to do the strategic planning. Up until about 2015, they could just do it and they felt like they could just lead and handle all of genomics. But the problem is that now genomics touches everything. And so it's almost impossible to strategically plan for genomics, that you really have to have a more limited approach. It's why the Institute rebranded itself, and it really represents a recognition that it can't be all about genomics, it has to be about the forefront of genomics. And that really served as an organizing principle when we put together this strategic vision, which is the latest one that came out in 2020. And you can even notice we even put our organizational mantra in the headline, or in the title of it. I would really, by the way, I say this every year, and it's actually not, it's really probably a good idea. I don't know if you've read this, it's 10 pages. It doesn't matter if you've read it. As a first year graduate, just go reread it. You know, maybe don't do it when you have exams. You know, you must have it when exams are over, I don't know what your schedule is. When you have time, you know, it's pleasure reading. It would be a really good exercise if you haven't read it lately, because what I find is when trainees read this, and they read it fresh, it gives them this newer context. All of a sudden, you'll read a paragraph, hey, that's what I'm working on. Oh, I never saw it that way, and you see contextual. So I don't know, at some point when you have a little bit of time, you might find it inspirational to reread it, because it may contextualize either something you learned in this class or something that you're working on. So I'm going to quickly skip through a couple slides, because it's not, I had a video, but I don't think that video is going to work. It's not going to work, so I'm going to skip through that. It's not going to work with the Zoom going. So what I just want to say in the last few minutes is a little bit of a wrap-up. What I would say, if you look over the course of my career, and in particular, if you look at what's happening in genomics today, is that, and you look back, is really this incredible growth of knowledge about, first we had the double helix knowledge. The Genome Project brought a nice magnifying glass increasingly to the sequence and to the functional aspects of the genome. And ever since then, the last 22 years, we've used the threads of the genome, if you will, for weaving together applications, first in more basic science and evolutionary biology, increasingly in human genetics, and then increasingly into genomic medicine. And now we just have more and more disciplines really helping to weave this all together. And we see more and more healthcare professionals getting involved in genomics, and it's all about making it mainstream. And mainstream is, I think, the real thing we need to be pushing for, especially thinking about some of the health equity issues and so on and so forth. So, last thing I would say is all this was going great for me. I was helping stimulate the weaving going on, but then came January 20th. So, the elephant in the room, if I'm giving a lecture, is, well, what happened on January 20th? Well, first this happened, and then this happened. And then you may say, how did you end up not having your job as NHGRI director? So I just thought I would tell you quite candidly that I drew a short straw, because if you're an institute, if you're an institute director at NIH, you are appointed on 5-year terms. And then you get reviewed, and if you're reviewed well, you get reappointed. So I drew the short straw that I had the first reappointment date in the Trump administration of March 17th. And I actually was reviewed back in December, and all the paperwork was done to renew me. But shortly after he came into office, the decision was made. They were interested in getting rid of some institute directors, and the easiest way to get rid of them is to just not renew them. So at the end of the day, they didn't renew me. It was a little weird how it happened, because apparently he was fine with renewing me, and so was the acting NIH director, but somebody else, like Doge or somebody, stepped in and they didn't want the institute directors renewed. So I was just not renewed, so I retired for federal service. I'll be fine, don't worry about me. But what I will tell you, so I sometimes joke that I fell into Doge the bullet, and this was some of the stories that came out, because I was the first institute director to lose their job in the Trump administration.
What an honor.
What an honor. It's not a bad honor, actually. Well, and so I take it with a little bit of humor. I was very proud. I was, remember this guy? But remember this guy. So I was the first NIH Institute director hired during the Obama administration. I was very proud of that. And now we have presidents who don't smile. And I was the first Institute director fired during the Trump administration. So okay, that's the bookend. People were really wonderful to me. I got a lot of nice emails and texts. People feeling very bad I lost my job. And I felt bad that they felt so bad. I have to admit, my favorite was a note that arrived, a little card. This was the card. I just think that's great. In any case, you have to sort of learn how to laugh on these things. What I would say is, I am wondering what is actually the point of treating NIH this way. I think it's really a shame. Here they talked about what happened on April 1st, a couple weeks after I lost my position. 4 and now 5 more institute directors have been terminated. They were not having to do with their reappointment date. They were just kicked out for personal reasons, or they didn't like the science going on at their institute. So that was much more political. Mine was just a convenient way to get rid of one. And all this is in the name of improving government efficiency. I can tell you stories that places not more efficient, it's much less efficient. They want radical transparency, and yet no institutes are updating their web pages because all their communication staff have been fired, including all of NHGRI's communication staff has been fired, so you can't even get things out because there's no transparency. And I do not believe that some of the scientific decisions are going to make America healthy again. Although, if we get a complete answer to autism by September, that'll be pretty good. I think that's about as likely to happen as me flying to the moon. But in any case, I think all this is very damaging. But I don't want to end on a negative note, because what I would tell you is that we should appreciate what an incredible field of science that we are now all working in. And we should recognize the trajectory we are on. It was a small community back when the Genome Project began, and I would say as much as anything else associated with genomics, I'm just so proud of this field. I mean, there's this idea of people standing on each other's shoulders, and now beginning to realize making differences in people's lives. I mean, we're saving lives. I mean, we truly are saving lives, we're improving lives, and that's happened over a relatively short period of time. And so we just need to ignore the politics, persevere, and keep building up this remarkable community of people who are doing dedicated and important things. So I will stop there, and it looks like I left lots of time for questions. So thanks. What's on your mind? Actually, while you're thinking of that, peers. Age peers, anything from history that you want to correct or augment?
Well,
because there's so much interesting history in all
I'll
this.
just ask, you said at the very beginning that there were two, I interpreted what you said as two fields or two areas that were crucial for the Genome Project. But you didn't.
Oh, I just meant that, I mean the very first slide, the genetics to genomics?
Yeah.
I just made that point that I just think there obviously other fields came into play. My main point was, I think a lot of people, especially younger, a lot of people just conflate the two fields, thinking that, they're basically the same field, and they came about the same time. I always like to point out they're very different. They're different. Those really, and there's a lot of interdigitation between them. Mm-hmm. And I think in many ways, the fact that genetics came into being in the early part of last century, now it's really bearing medical fruit through its merger with genomics or its synergy with genomics. Roger?
I would just say in the whole resistance to the Genome Project at first, certainly that resistance did melt away, but I think Shirley Tillman kind of captured that. So as this data started coming out, people could look in this database wherever they wanted to, and suddenly the gene you had been trying to find, or the genome region in which you knew that gene existed, was online. And you could search that, and you could find that, and the process of positional cloning of a gene, which could take forever, suddenly turned into just a quick search on the computer. And Shirley Tillman made a comment to the effect that the conversion of somebody who was against the Human Genome Project to somebody who was for it occurred almost instantaneously when they found their gene in that database.
I think that's a good one. It is fun to talk. I mean, I do talk to people all the time, and they'll say, "Man, I was originally against the Genome Project. Boy, was I stupid." You know, I always get these very funny comments from people who admit to being against it. I just didn't get it. Questions?
I think
Yeah.
a lot of the opposition, including Marty, and also reflected in things, something that Barton Childs used to say, is that genomics in the beginning was basically natural history.
Yeah.
And a lot of people thought science should be based on asking questions
Yes.
and answering the questions. And Barton's argument was that you should keep track of the sequences that are arising from people asking questions, and somebody should just keep putting them together.
Yeah.
But
Stitch
what
it.
you miss then is
What you don't know.
98% of the genome.
But you don't know. Right. Questions? Yes.
So I was curious about your point about diagnostic odysseys, and thank you, appreciate that. About diagnostic odysseys,
Yeah.
just because I think about some of the examples in my own family and
Yeah.
my wife's family. What do you think it'll take to sort of convince insurance companies to more front-load genetic testing? Because it feels like, we spend so much money
I agree.
sending people through years of other blood tests and things, and sometimes all it really took was genetic testing up front.
Yep. No, it is a great question. You could even lead your question into a broader question that gets answered is, at what point do we just say this is going to be so general purpose that we should just sequence every newborn and have that genomic information available in their medical record forever? So unfortunately, it's going to have to come, you point out, it's the insurance guys, it's the payers. They're going to have to pay for it unless there's going to be some big national effort to just sort of get this done in a comprehensive way. They're going to have to see enough value. There are big attempts to do this. There's a whole undiagnosed diseases network supported by NIH where one of their goals was to try to make this more mainstream in academic medicine, and the uptake has been slower than anticipated, and the payers don't want to pay. I think it's going to be. I think the best data is going to come from other countries. So I do think that I've looked at the UK, and thank goodness the Undiagnosed Diseases Program has now federated around the world to many other countries, which means they're being done in many other in healthcare delivery systems. The UK, I think, is leading the way, where they're doing a lot of things that may show that it is cost effective to get this done at an earlier stage. But these diagnostic odysseys are outrageously expensive. We need to get to that tipping point, and I wouldn't be surprised at the end of the day if the influence is going to come from abroad to convince the funders here. Dave, you have a
Just
thought
an
about that?
anecdote to underline this. When I was in the early days of when I was the director of the Institute of Genetic Medicine, we looked at our clinics, and one of the healthcare plans that was the most obstructionist to paying for genetic testing was the Hopkins plan. So we convened meetings with the leaders of that group, which of course came from Hopkins. And it turned out the way they were deciding whether or not they should pay for a test was that they had a contract with a private group that was headed by a nurse that reviewed the literature and helped them decide whether they were going to pay for genetic tests. And I said, well, some people think the genetics program here at Hopkins is one of the leading programs in the world. Why wouldn't you ask us? And they were so afraid that we would spend money that they didn't have, or spend money that was not didn't meet their criteria for justification. They were very, very resistant to doing that. It is amazing.
And the problem is, even if you would have won that battle, it's just one battle.
Oh yeah,
Just the next hospital, the next medical center.
But then you'd have to get it put in the electronic patient record.
Right.
And they have to do this.
Yeah, this is such an adult table problem.
Do you think the ambiguity in interpretation from genomic sequencing results is an argument against funding widespread genomic sequencing for diseases?
Well, I think it's fair to say that you won't prove utility, which is what, in cost effectiveness, if there's too many incorrect diagnoses. So, yeah, in the long run, but at the same time, we can't be petrified and cautious. I mean, we've got to start taking a little bit of, push the envelope, is maybe what I would say, but with an eye towards, there are a lot of the people are monitoring, seeing is it worth it, how many diagnoses, how much money are you saving, et cetera, et cetera. So, but I mean, I think we all can appreciate that, we just need to get really good at this. This is so important.
If I could make just another remark on it, I think one of the things that will drive it is what I see right now is a rapid expansion of treatment of genetic causation of disease.
Sure.
So before it used to be, well, this is just an unfortunate kid and they'll have to live with this genetic disease, to all of a sudden now you can say, no, here is a treatment that actually could have prevented further deterioration.
Or family planning. I mean, there was always the family planning.
Yeah, well, family planning, it's not such a big selling issue.
It's not a good selling issue,
Yeah.
except for the families.
Yeah,
Yes.
Unless it's your family.
Yeah, unless it's your
Exactly.
family.
But I think that this, what we heard from Greg Newby a couple of days ago, there are now more than 1,000 clinical trials going on in gene therapy.
Sure.
It's going to change a lot of stuff.
Roger?
I share your surprise that pharmacogenomics hasn't gone farther. I wonder if the pharmacy industry has issues with this.
Well, Big Pharma has, I don't know, but it's been disappointing. Yeah, I think Big Pharma has not embraced it as much. I naively thought when I first started learning about this that they'd be able to resurrect drugs that maybe had failed, at some stage of development. And then if they would actually just get superimpose genomic information, maybe they'd be able to stratify and find it was actually a good drug in a subset. Just those stories haven't really come about. Yeah, no, I'm sorry. I mean, it's a no-brainer in the oncology space where they're matching, chemotherapeutic agents or whatever to tumor genomic profiles. But they exist. It's just even where it's like no-brainer. And actually, some countries are much better at this than we are. That's the other thing. But again, it relates to this single payer. Like in the UK, if they want to say nobody's going to get this medication without a pharmacovigilance, they just one person makes that decision, one body makes a decision, it gets dispersed. So we need more examples once again in places like the UK where it's proved to be cost effective.
I agree with your point about drugs that came through clinical trials and had side effects in 30% of patients, and therefore Big Pharma said we're just going to trash this drug.
Yeah.
Genetics, you should say, okay, well, it works for 70%.
Yeah. Can we figure out how?
Can we figure out what that group needs the medicine and the other 30% should avoid the medicine?
Yeah. Any last question? Oh, yeah.
So I was thinking from like a broader public perspective. I think about a conversation I had with my aunt, who was like very much like huge fan of RFK, very much like very skeptical of most scientific endeavors.
Yeah.
And I remember she asked me, she's very kind though, she asked me what I was doing for my program. And I was like, I'm studying human genetics and genomics. She paused for a second and she said, what does that mean? She's like, what do you do? Right? What is that? And I was like, I had to walk all the way back. I was like, okay, like it's heredity. It's like these units of heredity. Anyway, I guess my question is, do you feel like our public education system is keeping up with these innovations that we're having so we're able to communicate what we're able to do with genetics now? If someone doesn't have a PhD or even doesn't have like a college degree in STEM.
Yeah. There's so many ways to answer the question. I mean, what I would say is we clearly have a problem just around science literacy. And there's not a strong enough foundation in fundamental science literacy, which particularly makes people vulnerable to scientific misinformation. I am overwhelmingly biased that this needs to be fixed, but I can't even imagine fixing the problem around all of science, misinformation. The joke I sometimes say is, we don't really. You have to teach basic science about geology or genomics. I think genomics is going to be more relevant to people's lives. So I have always stayed fairly focused on thinking about genomic literacy. And the reason I could make the claim is because it's become part of the language with healthcare professionals. So one approach you could use with your aunt is to start off by, giving vivid examples where genomics, is absolutely relevant. start with cancer, start with prenatal, start with you have a child with something doesn't seem right. Let's peer into their DNA and see if we can nip it in the bud, kind of a thing. I think this is going to be so critical. And, I know I'm biased, but I mean this is going to be increasingly discussed by healthcare professionals, and we've got to get people at least not being scared of it.
So with this being now 5 years old, the strategic
Yeah.
plan, where would you say, if you were to implement new pieces to that now, what would you add or change?
It's a great question. It's no longer a problem for me. It's a problem for you.
I know. But you're so biased that I have to get...
No. I can so many answers I can give. Because one of the things they're going to have to reckon with is probably start another strategic planning process in the next couple of years. And it's going to be really interesting because I will tell you, when we did this, actually, it would be interesting. Reread. When you're reading the strategic vision, look at it through a MAGA lens. I mean, look at it through a Trump lens, because just imagine this whole thing was being written and talked about during the pandemic, during the George Floyd thing. I mean, there was a lot of social consciousness that and appropriately so, we talk a lot about core values, including having a very diverse genomics workforce. So it's going to be interesting. We go to do that. How much, whether what the administration is, whether it's still MAGA, whether it's it's going to be very interesting to see that. I would say things I think that would be different. I actually think it's probably all pretty good. There's a lot more around AI now than maybe then. I think there was an appreciation. I think there's a lot. There'd probably be a lot more could be said about what's happening in AI. I also think there's probably. I mean, I'm not even sure how much in that. We were talking spatial genomics and some of these other things about some of the. So I think there's a few areas. But I think if you read it, you see we covered it pretty well. I don't think there's been any great surprises that are things that would have to be heavily fortified. And the challenges certainly remain. There's so much we still need to know about how the genome works.
I think one of the great successes of NHGRI has been sort of sticking to this idea of publishing, working on, and then publishing a 10-year plan, which Eric is largely responsible for, I would say, very rigorously. Other institutes, as far as I know, don't do anything like this, and it keeps NHGRI really focused and really up to date, and it's been very valuable over the years, I think.
And I don't know if we'd be allowed to do it now. We'll have to see. There's so much control being imposed, whether you could go off and publish, or they try to standardize these. The NIH, it's 27 institutes and centers, and they pretty much all generally can do their own thing within guardrails. Guardrails are coming in. I'd be curious whether they would allow it to happen now. But we'll see. But maybe we'll wait till after this administration's over. And
I think it's been incredibly valuable.
no, I completely agree with you. All right. Well, thank you for your time. It was always fun. Oh, one more, please. Oh.
I guess I was going to ask, I guess my assumption's always been, the public was also very excited kind of following along the Human Genome Project. And, I guess, if that is true, do you feel like there are things that are, inherently different now to try to get people kind of more excited about kind of general science? And
So first thing is, I think the informed public was kind of paying attention. It wasn't quite like putting a person on the moon, where everybody was captivated. I mean, nobody was sitting there waiting for that last base pair to be put in. so I don't think there was the informed public. I would say that I do think that people who are paying attention now are paying attention either a little bit of consumer genomics. I mean, I think 23andMe. While they're having issues now, I mean, they were blanketing the airwaves for a while. Everybody was talking about that. I think ancestry testing has gotten people very excited. That's an innocent way to get them involved in genomics. And there's enough cool stories that are coming out, and enough things. Lots of people know people who are, pregnant and getting this non-invasive test. Sadly, people are knowing people who get cancer and getting this genomic analysis done, and people are hearing more and more about gene therapy. So I think in general, except for the distrust, appropriately so, around eugenics and making sure We have to be very conscious of certain communities. We're a little more wary about genomics. At least it's not like vaccines. We see what's happening in that world, and I don't think that you see that kind of negative feelings about genomics in general. So. All right. Good.
Last year, projects, more diligent
Eric, I just would say, you mentioned how many years you've been teaching in this course, despite an incredibly busy schedule, and so we hope you come back. But,
Well, I hope I get invited.
Yeah.
Yes, you will.
I would just say that we've been incredibly thankful for you to make this.
I've enjoyed this tremendously.
And I would like to tell you the nuts and bolts. I would send out emails to faculty to have them set up all their lectures. The first person to respond, and the one who virtually never argued about the date had to come another time, was Eric Reed. The people who had to walk across the street, I might go back and forth.
That still continues.
17 scheduled this year. 507 or 501? What is it?
But I'll walk you.
Okay, all right, yeah, and I'll see you over there.
Okay, great.